Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Chi-square test for linkage analysis. Table of content. Introduction to chi-square test. Purpose of the chi-square test. Why use the chi-square test? Basic steps example solved. Let's start with Introduction to chi-square test. The chi-square test is a statistical method used to determine whether there is a significant association between observed and expected frequencies in categorical data. It's widely applied across various fields, including genetics, social sciences, business, and more. Purpose of the chi-square test. Number 1. Testing independence or association. One of the primary uses of the chi-square test is to examine whether there is a relationship between two categorical variables. For example, in genetics, it can be used to test if there's an association between observed and expected offspring ratios in a genetic cross. Number 2. Goodness of fit. It assesses how well observed data fit a theoretical or expected distribution. For instance, in genetics, we might expect offspring to follow a specific ratio, for example, 3:1 for a monohybrid cross. The chi-square test helps us determine if observed frequencies deviate significantly from expected frequencies. Number 3. Hypothesis testing. It helps in evaluating hypotheses about the distribution of categorical variables in a population. The null hypothesis, H0, assumes no significant difference between observed and expected frequencies, while the alternative hypothesis, H1, suggests there is a significant difference. Why use the chi-square test? Non-parametric test. It doesn't require assumptions about the distribution of data, making it suitable for categorical data where assumptions of normality might not hold. Simple and versatile. Relatively easy to understand and apply, making it accessible for researchers and practitioners across disciplines. Detects relationships. It's effective in identifying relationships between categorical variables, helping researchers understand associations or patterns in data. Decision-making. Provides a clear decision criterion based on p-values for accepting or rejecting hypotheses about the distribution or association of categorical data. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Steps for chi-square analysis. 1. Formulate the null hypothesis, H0. 2. Collect and categorize data. 3. Calculate expected values. 4. Calculate the chi-square statistic. 5. Determine degrees of freedom, DF. 6. Interpret the chi-square value. 7. Make a decision. 1. Formulate the null hypothesis, H0. The null hypothesis states that there is no significant difference between the observed and expected data. Any deviation is due to chance. 2. Collect and categorize data. Organize your observed data into categories based on the expected ratio, for example, 3:1 9. 3:3:1. 3. Calculate expected values. 3. Determine the expected number of occurrences for each category based on the total number of observations and the expected ratio. 4. Calculate the chi-square statistic. Use the formula where O is the observed value and E is the expected value for each category. 5. Determine degrees of freedom, DF. Calculate the degrees of freedom as the number of categories minus 1, DF n 1. For example, for a 3:1 ratio, n equals to 2 categories, dominant and recessive, so DF equals to 2 minus 1 resulting to 1. For a 9:3:3:1 ratio, n equals to 4 categories, so DF equals to 4 minus 1 resulting to 3. 6. Interpret the chi-square value. Compare the calculated chi-square value to a critical value from the chi-square distribution table, or graph, corresponding to the calculated degrees of freedom and the chosen significance level. Commonly, p equals to 0.05. Graph method. Locate the chi-square value on the x-axis. Draw a vertical line up to the line representing the appropriate degrees of freedom. Draw a horizontal line to the y-axis to find the p-value. Let's see how to use a chi-square graph to find the p-value. Suppose you calculated a chi-square value of 0.53. Locate the chi-square value of 0.53 on the horizontal axis. Draw a vertical line from this point up to the curve. Next, draw a horizontal line from where it meets the curve to the vertical axis. If the p-value is 0.48, this means there's a 48% chance the observed differences are due to random variation. Since 0.48 is greater than 0.05, we fail to reject the null hypothesis, indicating no significant difference. Table method. Find the row for the appropriate degrees of freedom. Locate the column where the chi-square value fits to determine the p-value. Let's see how to use a chi-square table to determine the p-value and make decisions about the null hypothesis. Here is a chi-square table for selected values of degrees of freedom, df, and p-values. In this table, darker gray areas represent chi-square values that lead to a p-value of 0.05 or greater, justifying failure to reject the null hypothesis. Lighter gray areas indicate values leading to a p-value of less than 0.05, justifying rejection of the null hypothesis. For example, suppose you have a chi-square value of 0.53 with 1 degree of freedom. Locate 0.53 in the row for 1 degree of freedom. The corresponding p-value falls between 0.20 and 0.50. Since this p-value is greater than 0.05, we fail to reject the null hypothesis. This suggests that the observed differences could be due to random variation. By using the chi-square table, you can easily determine the p-value range and make informed decisions about your hypothesis testing. 7. Make a decision. Reject HJO. If the p-value is less than 0.05, the deviation is significant and the null hypothesis is rejected. Fail to reject HZO. If the p-value is greater than 0.05, the deviation is not significant, and the null hypothesis is not rejected. Key points. A low p-value, 0.05, means rejecting the null hypothesis, significant deviation. A high p-value, 0.05, means failing to reject the null hypothesis, deviation due to chance. By following these steps, you can systematically conduct chi-square analysis and interpret the results to evaluate your null hypothesis. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Now we will solve one example to understand how chi-square test is applied on linkage analysis, as well as to compare the observed results with the expected results based on Mendelian inheritance. In a study of the inheritance of 2 genes in pea plants, a researcher crossed a pure breeding plant with yellow round seeds, YYRR, to a pure breeding plant with green wrinkled seeds, YYRR. All of the F1 offspring had yellow round seeds, YYRR. The researcher then crossed the F to each other to produce an F2 generation. The results of the F2 generation are as follows. Phenotype number: yellow, round is 602. Yellow, wrinkled is 191. Green, round is 202. Green, wrinkled is 605, totaling to 1,600. Using the chi-square test, which of the following statements can be true? Option A, the genes are linked. Option B, the genes are close to each other. Option C, the genes are in the same chromosome. Option D, all options can be true. 1. Formulate the null hypothesis, H row. The null hypothesis states that the 2 genes assort independently according to Mendelian genetics, following a 9:3:3:1 ratio for a dihybrid cross. 2. Expected ratios and values. The expected phenotypic ratio for a dihybrid cross, YYRR XYYRR, is 9 by 3 by 3 by 1. Total F2 generation equal sign 1600. Expected numbers. Yellow, round. 9 by 16 multiplied by 1600 equals to 900. Yellow, wrinkled. 3 by 16 multiplied by 1600 equals to 300. Green, round. 3 by 16 multiplied by 1600 equals to 300. Green, wrinkled. 1 by 16 multiplied by 1600 equals to 100. 3. Observed data. Yellow, round. 602. Yellow, wrinkled. 191. Green, round. 202. Green, wrinkled. 605. 4. Calculate the chi-square statistic. By this formula, input data, where O is the observed frequency and E is the expected frequency. After this calculation, answer for chi-square is 2,720.53. 5. Degrees of freedom. For a dihybrid cross with 4 categories, degrees of freedom equal sign 4 minus 1 equal sign 3. 6. Interpret the chi-square value. Compare the calculated KH2 value, 2,720.53, with the critical value from the chi-square distribution table at degrees of freedom, 3 and a significance level equal sign 0.05. The critical value for significance level equal sign 0.05 is approximately 7.815. Chi-square test results. From our chi-square test, we found 1. Observed phenotypic ratios deviated significantly from the expected 9 by 3 by 3 by 1 ratio. 2. The calculated chi-square value, 2,720.53, is much larger than the critical value, 7.815, at 3 degrees of freedom and a 0.05 significance level. 3. This leads us to reject the null hypothesis that the genes assort independently. Interpretation of results. Independent assortment and expected ratios. 1. This indicates a significant deviation from the expected 9 by 3 by 3 by 1 ratio. 2. Our observed numbers, 602, 191, 202, 605, do not match this ratio, indicating that the genes do not assort independently. So, let's see what option is the correct one. Possible explanations. Given that the genes do not assort independently, there are a few possible biological explanations. Linked genes. Option A. Linked genes. Genes that are located close to each other on the same chromosome tend to be inherited together. This is because the chance of a crossover event occurring between them during meiosis is lower, which means they don't assort independently. The significant deviation from the 9 by 3 by 3 by 1 ratio suggests linkage, meaning the genes for seed color and seed shape might be linked on the same chromosome. Genes close to each other. Option B. Genes close to each other. If the genes are very close to each other on the same chromosome, the likelihood of recombination between them is even smaller. This would further reduce the chance of independent assortment, causing the observed phenotypic ratios to deviate significantly from expected ratios. The large chi-square value indicates a strong deviation, which is consistent with genes being close to each other on the chromosome. Genes on the same chromosome. Option C. Genes on the same chromosome. Genes on the same chromosome can be linked, especially if they are physically close, as already discussed. Even if they are not extremely close, being on the same chromosome increases the probability of linkage and non-independent assortment compared to genes on different chromosomes. The observation that the genes do not assort independently supports the idea that they are on the same chromosome. Why all options can be true. Linkage. Option A. The genes not assorting independently strongly suggests linkage. Close proximity, option B. The significant chi-square value indicates a high degree of linkage, which is more likely if the genes are close to each other on the chromosome. Same chromosome, option C. For the genes to be linked, they must be on the same chromosome. Since all these explanations are interrelated and supported by the chi-square test results, any of the following statements can be true. The genes are linked. The genes are close to each other. The genes are on the same chromosome. Therefore, option D, all options can be true, is the correct choice because the chi-square test results are consistent with all three explanations. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!